Hi guys, welcome back to my channel. My name is Ciara Jessica Giron if you are new here, and I'm here today with a new segment of my nervous system series, and this segment is called Communication Between Neurons. If you are interested in buying the notes that I've taken for this chapter in the OpenStax textbook, you can see the link below in the description box to register as one of my students. And you will not only have access to my notes, but you will also have access to personalized tests that I have created myself, and I will grade myself for a small price. Before we get into today's lecture, I wanted to give an example of how neurons communicate within my brain. And this example will involve the music application that is called Spotify. Spotify is one of my favorite music applications because it runs on an algorithm to compile playlists of music that I would enjoy listening to based on my statistics. I personally compile playlists of my own using the Spotify algorithm to give me new music, and one of my habits is that I never listen to the same playlist in the same order twice. My reasoning behind this is because I feel like if I listen to the same music over and over again in the same order, it feels like I'm beating down the same neural pathways in my brain over and over again. I'd call this phenomenon neural pathway burnout or oversaturation of neurons in the same pattern. And this phenomenon can be compared to radio playlists that overplay the same songs over and over again to the point where I get annoyed with those good songs because their novelty has been wiped away. And so, to combat this neural pathway burnout, I like to listen to my playlists in a different order every single time. Sometimes I'll use the randomization feature on Spotify, and sometimes I'll just handpick all of the songs that I want to listen to at that moment. That's it for my food for thought. Now, let's get into a recap of what we learned in last lecture. In my last lecture, we talked about the nature of electrophysiology as it pertains to electrical impulses within neurons. We talked about how transmembrane proteins exist to transport ions across the cell membrane to keep concentration gradients constant at resting membrane potential. We also learned about how to read the action potential graph as it illustrates the events that happen within a neuron over time to conduct electrical impulses. Finally, we talked about how the myelination of axons in neurons affects the speed at which electrical impulses are conducted. Now that we know how electrical impulses are conducted via cell depolarization, our next question is, how do neurons initiate communication with each other? Could it be another nearby neuron's activity? Or could it be the environment? Special types of transient signals that influence whether an action potential will occur or not are called graded potentials. These graded potentials can either be depolarizing or hyperpolarizing. There are several different types of graded potentials. For example, a unipolar neuron develops potentials within that neuron's dendrites and eventually results in an action potential that propagates down that same neuron's axon. This is called a generator potential. In other sensory neurons, like taste or photoreceptor cells within the retina, graded potentials result in the release of neurotransmitters on postsynaptic sensory neurons. This is called receptor potential. It is important to remember that generator potentials result in action potentials, while receptor potentials result in neurotransmitter release. What is postsynaptic potential? Postsynaptic potential refers to the graded potential that a target neuron's dendrites receive from a presynaptic neuron. What is an excitatory postsynaptic potential? An excitatory postsynaptic potential is a depolarizing graded potential that moves the membrane potential up towards threshold. What is an inhibitory postsynaptic potential? An inhibitory postsynaptic potential, or IPSP, is a hyperpolarizing graded potential that moves the membrane potential away from threshold. What is summation? Summation refers to the way that small graded potentials, both excitatory and inhibitory, can be combined or summated to result in an overall depolarization or hyperpolarization of the membrane potential. See this figure. If the total change in membrane voltage reaches above 15 millivolts, an action potential will be observed. Spatial summation refers to the result of multiple graded potentials applied at different locations on the neuron. Temporal summation refers to multiple graded potentials occurring at the same place on a neuron but at different times. What kind of synapses exist? There are electrical synapses which occur between neurons that are physically attached to each other and therefore share depolarization events. There are also chemical synapses. Chemical synapses involve neurotransmitters, also known as chemical signals, that are released from presynaptic cells onto postsynaptic cells. In this lecture, we will be focusing on the chemical synapses. For neurotransmitter systems, you can read in great detail in this chapter about the different neurotransmitters that exist within the nervous system. In this section, I will be talking about a specific neurotransmitter system that distinguishes receptor types. It is key knowledge to know that the effects of the neurotransmitters are entirely dependent on the nature of the receptors that receive those neurotransmitters. What are ionotropic receptors versus metabotropic receptors? Ionotropic receptors are ligand-gated ion channels, while metabotropic receptors involve a series of proteins that affect the metabolic changes within a cell. In the first figure, A, you can see that a neurotransmitter binds a ligand-gated ion channel, otherwise known as an ionotropic channel. You can see that an immediate response is observed as the neurotransmitter triggers the opening of the channel to admit ions into the cytosol. In the second figure, B, it gets a little more complicated with metabotropic receptors. First, the neurotransmitter binds to the metabotropic receptor. This event attracts the G protein to the protein complex, where it meets the effector protein. The effector protein cleaves ATP to generate a second messenger, cyclic AMP or cAMP, and this cyclic AMP can go on to change the cell's metabolism, gene transcription, or open and close ion channels. As you can see, the chemical signaling that occurs between neurons establishes a highly variable system through which neurons can affect each other. Back to my music listening pattern, perhaps the joy that I feel whenever I listen to my music in varying patterns is owed to the fact that my neurons are communicating in symphonies that always sound new. New combinations of sound breed new auditory sensations, which eventually lead to new perspectives on life and about the music that I am listening to. As you can probably tell, I'm obsessed with music and I love listening to music as a coping skill to keep my mind healthy and active. Anyway, thank you so much for making it to the end of this video. I hope I taught you something about communication between neurons that might help you on a future test. And if you are interested in becoming one of my regular students, you can register as one of my students using the Google link that I've put in the description box. Again, thank you for watching. I'll see you in my next video. Bye.